Okay, perfect. Good morning, everybody. My name is Melania Munoz, and I work at the DSMC, the German Collection of Microorganisms and Cell Cultures in Germany. And I am here today to talk about the Nagoya Protocol and what does it mean in practice for collections and for research. So today we will cover the basics of the Nagoya Protocol, what are the obligations under the European Union Access and Benefit Sharing Regulation, and some tips for ABS multi-partner, multi-country projects that involve the use of genetic resources. So let's start with the basics of the Nagoya Protocol. It is not a secret for anybody that the biodiversity is decreasing in all continents, and this biodiversity loss is causing important consequences for our day-to-day lives. So in order to face this problem, in 1992, the United Nations adopted the Convention on Biological Diversity with three main objectives, which are the conservation of biological diversity, its sustainable use, and benefit sharing from the use of genetic resources. And to implement the third objectives about benefit sharing, the United Nations also negotiated the Nagoya Protocol. And it was adopted, or it entered into force 12th of October of 2014. And this is a comprehensive international framework for access and benefit sharing and compliance. But what is ABS about? ABS comes from access and benefit sharing, and access means taking and using biological material for research and development and asking for permission first. And benefit sharing is giving something back. It could be many things like sharing research results, joint research and publication, training, in-kind support, and other kind of monetary and non-monetary benefits. And it should contribute to conservation and sustainable use of biodiversity. The most important pillars of the Nagoya Protocol are the following. Number one, access to genetic resources. Each country decides what rules they will implement to regulate access to their own genetic resources. In the case of the European Union, this is not implemented at the EU level. So each member state has the right to decide if they regulate access to their genetic resources or not. So, for example, Germany decided to do not regulate access to their genetic resources, but other member states, as France or Spain, they do regulate access to their genetic resources. So you need a permit in advance. Pillar number two is benefit sharing. And the idea here is that the user and provider of these genetic resources sign a contract and they negotiate what kind of benefits the user will share with the provider country. And the pillar number three is compliance, and compliance is regulated at the EU level with the EU ABS regulation 511 from 2014. And this regulation established the due diligence obligations for all users of genetic resources. And I will explain more about the EU regulation in a moment. But why the Nagoya Protocol is important? Access and benefit sharing measures in the different countries ensure that research results support provider countries to many different things. For example, to make informed decisions on conservation, sustainable use, and management of their own biodiversity. So information is important for provider countries. Then implement innovative solutions for productive activities, including, of course, agriculture, and contributing to sustainable development. Build capacities for local research to solve local problems is pretty important, and also other monetary and non-monetary benefits that could be agreed in these ABS contracts. And what are the obligations under the EU ABS regulation? I will explain this for collections and for research. So as you may know, the SMC houses the most diverse collection of microorganisms in the world. And we receive around 2,500 deposits per year, and we distribute around 43,000 bioresources per year to more than 10,000 customers in more than 86 countries all around the world. So as you see, we exchange a lot of bioresources, and that's why the SMC must fulfill all the international obligations for exchanging biological material. It is important to mention that the mere storage of biological material without conducting any research is out of the scope of the EU ABS regulation. So just storage in material is out of the scope, but if within the collection you have some research, the researchers must be aware about the Nagoya Protocol and request of all the necessary permits. Other thing that could impact the Nagoya Protocol is the description of new species, because according to the International Code of Nomenclature of Prokaryotes, for description of new species, you must deposit these new strains into different collections without restrictions for redistributing. So if the Nagoya Protocol permits you obtain to collect and use the microorganisms, establish some kind of restrictions for distribution, you could have problems when you try to deposit these strains for the description of new species. And this is especially important for some countries, for example, for India and for Brazil. Then the EU ABS regulation also established a voluntary compliance mechanism that is the register of collections. So there is an official mechanism for registered collections under the EU ABS regulation, and this procedure is optional, it's not obligatory. But the Leibniz Institute, the SMC, decided to go through the process and became the first registered collection under the EU ABS regulation. It was a long process, 2.5 years process from November 2015 to May 2018. And we have to redesign all the accession forms. We have to create the catalog, and then we have to publish this catalog update. So if you go to the DSMC catalog and you would like to obtain some of the strains, you will have a very clear information about the Nagoya Protocol restrictions. So if the strain doesn't have any restriction, it will be very clearly specified there in the catalog. And on the other hand, if the restriction does have any restriction, if the strain does have some restrictions, it will also be specified. So it is important because the user or the customer of the DSMC strains, they will obtain all the information they need from the catalog, and they will obtain all the documentation they need. So it was a very long process, but it has also very important outcomes. For example, users can easily fulfill their Nagoya Protocol due diligence requirements. Also, it increases the usability and legal certainty of the DSMC collection. And because we acquired a lot of experience with the Nagoya Protocol implementation and compliance, we had a positive domino effect on extramural funding for projects related to ABS. So it was a positive process at the end. Then for research, the Nagoya Protocol is relevant for you if you do research with genetic resources or derivatives. So what is a genetic resources according to the Nagoya Protocol? So it is any biological material that contains DNA or RNA, even if it is dead or alive. For example, if you take some samples from a herbarium, or you do like fossil excavations and you obtain DNA from those samples, this is also within the scope of the Nagoya Protocol. There are some exceptions. So for example, the Nagoya Protocol excludes human DNA. So if you work with some hereditary diseases in humans, this is out of the scope. But if you study human pathogens or human microbiome, this is within the scope of the Nagoya Protocol. Another exception are plant genetic resources or the International Treaty for Plant Genetic Resources for Food and Agriculture. This international treaty has an annex, and all the species included in the annex are excluded for the Nagoya Protocol. Then the influenza strains under the pandemic influenza preparedness framework are also excluded from the Nagoya Protocol. So in the case of the plant treaty and the PIP framework, the material and use must be under the scope of these agreements. For example, if you use rice for food and agriculture research, then it is out of the scope of the Nagoya Protocol. But if you use the same rice for developing some medicines or cosmetics, then this is out of the scope of the plant treaty and is within the scope of the Nagoya Protocol. So it is important to notice that. And then it is not just studying DNA and RNA, but also what they call derivatives. And derivatives are naturally occurring biochemical compounds resulting from genetic expression or the metabolism, even though it does not contain functional units of heredity. So some examples are proteins, including enzymes, lipids, organic compounds, and other products of metabolism. So metabolites are also within the scope of the Nagoya Protocol. And this is important. And then it's also important to mention that the ABS world has two parts and researchers need to understand both. So you can, if you visit the German Nagoya Protocol Hub website, you will see this infographic and we explain and we have some key questions you have to make to yourselves in order to see if you are within the scope of these two parts of the ABS world. The first part are the obligations according to the ABS rules or legislation in provider countries. So you first must check the ABS legislation in the country where you are obtaining the samples. And then the second part are the obligations under the European Union. If you are performing your research within the European Union, you have to check the EU ABS regulation to see if you are within the scopes and which are your obligations there. So obligations under the provider national rules and obligations under the EU ABS regulation. So whether you have compliance obligations in the European Union is determined by the regulation 511 from 2014. So if you, to check if you are within the scope, you have to answer the following questions. So question number one is, does the material contain DNA, RNA, or derivatives? Yes or no? Are you doing research on the genetic and or biochemical composition of the material? So this is important. This is the utilization scope under the EU regulation. And it is a bit tricky, so my recommendation is go through the guidance document, and we can share the link in the chat later. But the guidance document has a lot of very informative and easy to read examples of what is considered utilization under the EU ABS regulation and what kind of research is out of the scope or within the scope. So it's important to check. Then the third question is, was the material originally obtained on or after 12th of October 2014? This date is very important because it's when the Nagoya Protocol and the EU ABS regulation entered into force. So it regulates that. And the fourth question is the provider country a Nagoya Protocol party and has ABS measures in place at the time of access? Yes or not? So you have to check in the clearinghouse and you have to do your research to see if the provider country, the country where you obtain the samples, is party to the Nagoya Protocol and if they regulate access to the genetic resources. So if you answer yes to all these questions, you are within the scope of the EU ABS regulation. And it is important to highlight that the national ABS legislation may have different scope than the EU regulation. You have to check both. Because even though you are out of the scope of the EU regulation, you may have obligations under the provider country's national legislation, and you must comply with those national legislations too. So what are the obligations? The EU regulation established obligations for users of genetic resources. So users must comply with the ABS measures in provider countries. You have to check if that country regulate access to the genetic resources and which are the requirements, if you need a permit or what kind of documents you need to access their genetic resources. Then you have to seek and keep these permits. You have to keep these permits during 20 years after you stop utilization. And if you are transferring this material to other partners, or if you are depositing in some collections, the ABS information should go together with the samples. So you have to transfer this ABS permit to subsequent users. And then the last obligation is to exercise the due diligence declaration regarding legality of access. And this is done very easily in the EU Declare website. So you do it online. So those are your obligations. Also, the EU regulation established that the different member states should perform user checks to verify that the researchers and institutions comply with the EU ABS regulation. In the case of Germany, the Federal Agency for Nature Conservation, BfN, is the one in charge to perform these user checks. And then there are also implications of non-compliance. So, for example, in Germany, the sanctions could be fines up to 50,000 euros, orders to stop research, confiscation of material, publication embargo, or publication retractions. But there are also other kind of sanctions abroad established by the provider countries that come from fines to jail in some cases. And there are some other non-regulatory consequences of non-compliance, for example, loss of reputation, loss of trust in provider countries, and to be included in a blacklist of people or researchers that have accessed genetic resources illegally. And these two are some examples of publications that highlight some researchers doing some work with illegally collected genetic resources. So this is very serious, and you have to take it seriously and take care of the Nagoya Protocol if you don't want to have these consequences. And then some tips for multi-partner projects, how the multi-partner, multiple country projects implement ABS compliance. So we recommend to develop your ABS strategy. We have a guide in our website, step by step what you have to do, but it's very important to try to standardize some of the terms, especially, for example, what kind of benefits the project is willing to share with the provider countries. Then it's important to define ABS roles and responsibilities, who is responsible for what, and be sure that somebody is responsible to obtain the necessary permits and to store it. Then comply with the ABS obligations. That means approach the provider countries and obtain the necessary permits. Provide resources and training to be sure that every partner is aware and everybody is on the same page regarding to the Nagoya Protocol, and a good manage of ABS documentation. It is important to compile and to have accessible all the ABS permits and to have everything very well organized in case your institution is selected for a user check. And we, in the German Nagoya Protocol Hub project, we are building collaborations with projects. So if you need some help with ABS compliance, you can contact us and we could help. You can also visit our website. We have a lot of material that is translated from the legal vocabulary to easy to understand vocabulary. We have some interactive infographic, as I mentioned, the ABS word infographic, and we have some guidance as the ABS strategy guidance. And we also have a help desk that if you have more specific questions, we can also help you. And this is my presentation. Thank you very much for your attention.
Thanks a lot. if you prefer to take questions on this part and then we go to the second. I don't know.
Yeah, it could be for me. Yeah, it's okay.
Yeah. So I leave the floor to the people. I think there are many interesting aspects. So if anybody has got questions.
Good morning. I have one question.
Introduce yourself. No, where's the voice?
Yeah, where's the voice? Okay, the microphone is there. Okay. Okay. I just have one short question to start with. Can DSMZ be involved in Horizon project as partner? Is it possible for you to be partner of Horizon project?
Yeah, we have some Horizon projects in DSMC. One of these is microbe project that is working on collecting and saving and also developing some business strategies for microbiomes. And yeah, we have an important component in this project related to compliance with the Nagoya Protocol. So it is important. We have done some workshops and information sessions for the partners of the microbe project in order to everybody is aware of the Nagoya Protocol and to comply with the regulations. There should be, this is just the project we have in my department. There should be some other projects funded by Horizon Europe in the institution, of course.
Thank you, Francesco. Other questions? I have one. I'm Eduardo Puglisi. I'm quite familiar, not completely, but the part relating living cells, so microorganisms regarding Nagoya Protocol. And then it's very interesting what you mentioned about the DNA, RNA, and also metabolites, right? Because indeed, any kind of samples we work with will have DNA or RNA inside, right? And soil and whatever is almost impossible to find without. So my question is, because in the case of microorganism, it's pretty straightforward. I find a microbe of interest, I have a plan to somehow exploit it, right? And then, of course, I must comply to the Nagoya Protocol, right? But regarding DNA, RNA, of course, any study we got will probably very often involve the DNA sequencing and so on. But when it starts to be applied, the Nagoya Protocol, I guess, I don't know, maybe if the sequence of DNA I find in a certain soil has got a biotechnological application, then this is the case, I guess. Are there other relevant cases? Because many of us work with metagenomics and so on, so I think this is very important for us to know. Thank you.
Mm-hmm. Yeah, I think, actually, this DNA, RNA, and derivatives is super general. That's why I mentioned the utilization scope. So, for example, if you do just comparison, and if you like, for example, 16S, or if you sequence the genome, but you do not study the composition of the genetic, if you don't do research on the genetic composition, so you are out of the scope of the EU regulation. So, for example, if you have environmental DNA and you would like to know what species you have there, and then you run the 16S or any other kind of identification technique, this is out of the scope of the EU regulation. But then if you study the composition, if you study the function of the genes, the expression of the genes, if you study what kind of proteins are produced by a specific genes, so this is studying the genetic composition, and this is considered utilization under the EU regulation. So this is the tricky part sometimes, and as I mentioned in the guidance document for the EU ABS regulation, I will copy, when I finish the questions part, I will copy the link in the chat. Then you can see specific examples, and then you can compare these examples with your research, and you can figure it out if you are within the scope or out of the scope of the EU regulation. But keep in mind that even though you are out of the scope of the EU regulation, you may be within the scope of the national legislations. For example, many of the national legislations, they do consider taxonomy or identification of species within their national legislation. So even though you are out of the scope of the EU regulation, you must comply with the national legislation of the provider countries, with the countries you are taking the samples. So it is important, and this is the complexity of the Nagoya Protocol, that every country has different measures, so every country has different scopes. So you really need to check each country's legislation or information to check if you are within the scope of some of these regulations.
Just a short heading on this, but for example, if I sequence DNA, I do a metagenome, and then very often these sequence, including functional genes, are uploaded on the NCBI, and they are public. So this means that if somebody goes into this metagenome, found the genes of interest, and exploited, must comply to Nagoya as well.
Yeah, it depends on the country where you obtain the sample. But yes, for example, if you just sequence the genome and upload it in the gene bank or INSDC, then you may be out of the scope of the EU regulation. But if you, for example, annotate the genome and you said, well, this gene produced this enzyme and this gene produced this protein and so on, then you are within the scope because you are doing research on the genetic composition of this genome. But even though you do not annotate the sequence, if you just sequence and publish the sequence, you may be within the scope of the national legislation, even though you are out of the scope of the EU regulation. That's why it's very important to have clarity about these two parts of the ABS work. Like say, you first have to check the national legislation and see if you have to comply with something, and then check the EU regulation and check if you have to comply with something. But yeah, it could be different depending of the country where you obtain the samples.
Okay, thank you. We have other questions?
There is a question from Georgia. Hello, everyone. Good morning. Have a nice week. Melania, thank you for this presentation. It is always very useful to observe this kind of issues. I'm Georgios Karakatsanis from Evotropi, and we're dealing in SIRAM with economic issues. So eventually I had only one question, but with the discussion with Eduardo, there came up a new one. You mentioned the distinction between the European legislation, the framework, and the national legislation. What happens in case you have contradictory or non-compatible, let's say, legislations between a state and the European Union? For the European Union's countries, in general, although it might get a little bit more complex, the rule of thumb is usually that the European Union's legislation prevails. So the national legislation usually has to comply with the European one. However, this is not something very strict. Have you met any cases where the national and the European legislation are contradictory, and what happens in that case?
Okay, there are different things. The EU ABS regulation is focused on compliance. So what the EU regulation says is that this is the scope of the EU, so you must check the temporal scope when you collected the samples, where the samples come from, if this country is party to the Nagoya Protocol, and if they have legislation, if you are under the utilization scope, and if you are using genetic DNA, RNA, or derivatives. So the EU legislation is on compliance, so every single EU member state must establish user checks and see if the users within their jurisdiction are complying with the regulations established in provider countries. So this is the EU. Then each member state can decide if they regulate access to the genetic resources or not. So they regulate the national legislation in case of the EU member states is on access to the genetic resources. So for example, Spain and France and Greece, they have rules, they have specific procedures that researchers must comply to obtain a permit, and they can legally access genetic resources from these countries. So the scope of these legislations are a bit different because the EU legislation is about compliance, and the access legislation in each member state is about rules for access. So you must comply with the rules of access, and then you need to do your due diligence declarations under the EU member and the EU regulation. So the member states can have access regulations that could be more or less how do you say, they could have more things within the scope, and this is not a problem because the EU regulation do not establish restrictions of what countries can do with their own genetic resources. So that's why it is important to check the national legislation. There are not contradictions, just to clarify and concretely your question, there are not contradictions between the EU member states' national legislations and the EU legislation. Then for countries outside the European Union, they also regulate access to genetic resources. For example, in America, in Africa, each country has the sovereignty right to establish the rules they consider appropriate. So there is no contradiction. The only thing you have to check is that the scopes are different. So you have to check the scope of the national legislations to see if you have an obligation with that country that you must obtain a permit. And then you have to check the EU ABS regulation to see if you have obligations under the EU. And this is related to compliance, and this is related to the due diligence declaration, that you have to declare that you obtain the genetic resources legally from the provider.
Okay, if I understand well, so it is something like complementary, and the national legislation works a little bit at the secondary level to complement and make more specific rules. And okay, the main question that I had from the beginning was the following: perhaps if you have any idea, or if you have encountered the case, a very significant issue currently regarding the utilization of generally genetic resources and ecosystems has to do with environment, society, and governance criteria, the very well-known ESG, and this is, let's say, the cornerstone of the European Sustainability Finance Taxonomy. Are there any specific regulations in the EU framework that make it clear, in order to finance such investments in the utilization of genetic resources, that it is obligatory to follow specific rules? Is it something that you have encountered the case? I suppose that it is something unexplored currently, but eventually this will be heavily discussed in the near future.
Okay, I didn't understand really well the question, but you're saying that if there is some general regulation for commercialization of products developed using genetic resources?
An investment framework, but regarding in compliance, that mentions specifically that has to be in compliance to the European Sustainability Finance Taxonomy. The taxonomy is a very hot issue currently. I mean, regarding the European Green Deal. If you have something in mind that is currently ongoing on this one for an investment framework that includes the Nagoya Protocol, ESG criteria, and the European Sustainability Finance Taxonomy, just in case?
Really, well, now we are really focusing on non-commercial use of genetic resources. So we help non-commercial users. We help universities and research institutions with their obligations under the Nagoya Protocol. And we don't really have some engagement, like strong engagements with the commercial users. There are some, of course, there are restrictions, or there are obligations for commercial users under the EU ABS regulation, but then I don't really know the other framework you mentioned. So for commercial users of genetic resources, you must do your EU, the due diligence declaration in the declare system under the EU for the research part. And then if you want to commercialize the product, you must do a second due diligence declaration under the declare before you commercialize your products. So of course, the Nagoya Protocol have implications on commercialization of products developed using genetic resources, but yeah, this is the same EU ABS regulation. So you must take care of that. But we don't have any other further work related to commercialization of products.
Okay, thank you very much, Melania. Very useful presentation.
You're welcome.
We got Lobna with a question, please. Hey, Lobna, you got the microphone closed. No. Okay. Otherwise, we can move on with the. There's somebody else with questions? Okay, so maybe we can move to Felicitas. Yes. For the second
Thank you very much for your attention.
It's been very, very useful. I think we could discuss for half an hour or 1 hour, even more.
I will share with you the presentation, then you can distribute it, and I will share in the chat the project website and also some interesting documents that could be helpful. Thank you.
We introduce Felicitas Bajerski from DSMZ as well, who's going to probably talk a little bit more about the process necessary for safe and fair deposit of microorganisms on international DSMZ. Thank you.
Yeah, good morning. I hope you can hear me.
Yes.
Can you? Yes, perfect. Then I will share my screen. Can you see the presentation?
Yes.
Perfect. Okay, so thank you for this kind introduction and for the invitation to speak about safe and fair deposit of microorganisms. And Melania already gave a very nice overview on the Nagoya Protocol. And now I will refer to the different kinds of deposit and also tell you something about ownership and also patent system and the Budapest Treaty. So first, as a small introduction, the DSMZ is both a research infrastructure. So we have the active public collection for that is used by science and also by industry for non-commercial purposes. And we also a research institution where we do active research on biodiversity. And we are one of the most diverse biological research centers worldwide, and also one of the largest collection of microorganisms in cell cultures. And as Melania already told, the DSMZ was the first registered collection under the EU regulation for the Nagoya Protocol. And my part will more focus on like the other side, on the more restricted part. So we are also in Germany, the only recognized international depository authority under the Budapest Treaty. Yeah, so at the DSMZ, we have 3 different kind of deposits. So, and this is also common for several other microbial collections. So we have the public collection that is available for the international scientific community, where we have biological material deposited that is of interest for applications, for biotechnology, for education and taxonomic research. So it is of general interest, and you can deposit your organisms here if work is already published or if you're about to publish some work. And this is also all in the obligation of open science. And yeah, as I just mentioned, and as we heard before, the DSMZ is also a registered collection, and so the strains you can buy at the public collection, they are also like Nagoya compliant, so we provide all the necessary information that are necessary for you due diligence. And besides the public collection, we also have a restricted collection where we provide safe storage and controlled access to the biological material. So on the one hand, we have the safe deposit where everything is handled strictly confidentially, where the material is only available for the depositor, but where the deposit is also not suitable for other purposes such as scientific publications or patent purposes. And on the other side, we also offer the patent deposit according to the Budapest Treaty, and this deposit is internationally recognized. And also here the material is strictly confidentially. And in the following, I will tell you a bit more about also ownership and property rights, and go deeper into the Budapest Treaty system and patent deposits. So, but first, some definitions we have from law, because this is not so easy to divide. So we have ownership, and this is a state or legal fact of possession and control over a property. And this ownership can involve multiple rights. So it's not just one law, but multiple laws that regulate ownership. And this like ownership can also be separated to different stakeholders, and this can be held by different parties. And the possession is a control a person intentionally do over a thing like a property, and like ownership, the possession is regulated by property law. And to possess something, a person must have the intention to possess it, and also the intention to use it, and have control over it. And like to understand the different, you can say that a person may be in possession of something, but without being the owner of the thing. So this is really different situations also under law. And property is a system of rights, and usually it involves different laws and gives legal control about things. And here usually we have defined the properly use of things. So it's not the ownership, but it's how an owner can use a property, a thing, under granted property rights. So for example, you consume, you can share, you can destroy, you can rent, you can sell, and you can also exclude others from using things. So that is all regulated by property law. And later, when we come to patents, is also intellectual property law. Because the general question, who owns a microorganism or biological material, is not so easy to answer, because it's not really clearly regulated by law. But we have different property laws and also other laws that refer to this kind of situation. So usually you define rights of use. So for example, property rights. And for the DSMZ terms of use, when it comes to the public collection, for example, we exclude any commercial use of the material. It is only used for research purposes. It is not allowed to distribute it to third parties. And of course, you also have to be like Nagoya compliant. And for future publication, we are asked to give credit to the DSMZ collection. This is completely different for the patent deposit, for example. Here we rely to different laws that are set up by countries or also EU, for example. And for European patents, it is regulated that the material you can receive, like that is mentioned in patents, and that is deposited for patent purposes. This is only used for experimental purposes, and it's also not allowed to transfer to third parties. So this is like if you apply for the use of a material, then you have to sign such a form. So so what is a patent and what is an invention? So first we have the invention, the idea, the intellectual property. And this is usually a product or a process, like a new way of doing things, or also a new technological solution for a thing. And this and the patent gives exclusive right about this invention, about your idea, or about your new, for example, machine or a new enzyme you discovered. And the patent, on the one side, gives right to the inventor, like it gives legal protection for the invention, but the patent system also benefits the society because it gives access to the invention, so to the technical information that people can try to rebuild this this invention, of course, without interfering with the patent laws. And what is actually patented when we come to microorganisms? So usually it is a process by which biological material is produced or processed, or which it is used. And usually, at least for for example, for Germany and also for the EU, it's not the material itself, not the strain itself, but something a strain, for example, produces, or where you can use the strain for. But this regulation can also be different in different countries. And like every patent needs to be new or novel and non-obvious, and it has to be needs a useful purpose, so also a commercially usable application, and it must be disclosed. So and in a way that an expert in the field can carry out the invention. So usually, if we would have a machine or an idea, we would maybe hand in a drawing, like a plan. But when it comes to microorganisms, it's not so easy, or like it's not possible to rebuild a microorganism, and that's why you need to another way needs to be found to guarantee that this invention can be also used. And therefore, it was decided that disclosure can be done by depositing the microorganism in a specialized institution. So you can't also just send your microorganism to the patent office because they are not a laboratory and couldn't organize this. So in 1977, the Budapest Treaty on the national recognition of the deposit of microorganisms for the purposes of patent procedure was implemented. And and this treaty was adopted in 1977, and already in the beginning there were almost 90 members, but it's also the contracting parties, they are still growing. For example, the last accession was Rwanda in 2023. And what is very important and also interesting is that the Budapest Treaty is also ratified by the big global players such as China, Russia, and the US. So it's really a global system and really important. And I checked the newest updates on the treaty, and just recently also there is a new International Depository Authority in Portugal. I guess some colleagues in the audience are also here. So yeah, it's always nice to get to know some colleagues. And overall, we have 51 depository institutions in over 27 countries. And the main task of the IDAs, which is a shortcut, is the deposit of microorganisms and also the furnishing of microbial samples under the Budapest Treaty. And yeah, here I collected some main facts, because the interesting, or like the main goal of the treaty was that one single deposit in one IDA is accepted by all partners, or by all states that ratified the Budapest Treaty. So if you want to apply for a patent in the US, and maybe also in China, it is just necessary to have one deposit, and this can also be in Germany, for example. Yeah, so you don't have to put your microorganisms in all the countries where you apply for a patent. And the storage is of the microorganisms has to be for at least 30 years, to cover all the patent period. And this deposit also remains valid, so you can't cancel this deposit. And the material is deposited with the IDA, but it is also necessary that the depositor keeps a backup stock if something happens with the IDA, or with the sample of the IDA at the IDA. And yeah, of course, the depository authority remains secrecy, and the material and also deposit is kept strictly confidentially. Yeah, so the DSMZ is a patent depository over 45 years, so even before the Budapest Treaty system, we have patent deposit. The first strain was deposited in 1972, and the DSMZ became an international depository authority in 1981, so shortly after the treaty was ratified. And since then, we have collected over 9,000 patent deposits. And yeah, you can see that the numbers are a bit fluctuating, but like we have about 200, 250 deposits per year, so the dark red bars, and we have between 50 and 200 samples that we furnish to third parties, or also to the depositor, of course. So that's my daily work. And we have different, and besides this patent deposit, we also offer the safe deposit. And here, this is a special long-term, like a special service for long-term preservation and safe storage, besides the deposits in the Budapest Treaty system, because there may be also other application where you need a safe and or like a backup deposit. And also those strains, they are not included in the catalog, and they are also only passed by written request of the depositor. And here we also have like about 200 deposits, and also about 100 samples that we give to our depositors. And this kind of deposit is usually used to maintain available biological resources for future research, but also for bioeconomy and public health. And yeah, it is also like this backup storage is used to overcome potential risk, such as pandemic situation, like that we experienced during the COVID-19. But this may also be other crises and disasters, like war or environmental problems. And like this backup storage can like the aim of this backup storage is to avoid loss of material made through staff shortage or infrastructure related supply supply problems and disasters. And yeah, and sometimes, or like in many cases, this backup storage is also mandatory for certain registration processes. And so, for example, our safe deposit is used quite frequently for the European safety, Food Safety Authority applications if you apply for feed additives, for example. So if I go back one slide here, you can see that in 2020, we had really an increase of deposit due to the pandemic situation because people want to have a safe backup for their samples. Yeah, and yeah, in the last few minutes, I will shortly guide you through our deposit process. So this is based for the, this is mainly concentrated on the patent deposit, but the general procedure is similar for the safe deposit. So we have an accession form, which is an international form where we collect taxonomic designation, identification reference, cultivation information, but also important information regarding risk assessment and genetic manipulation of the strain. And when everything is settled and we give green light, the depositor can send the material to us. And we have different biological material. And we have we accept risk group 1 and 2 organisms that may be also genetically changed up to safety level S1 or S2. And we have all different kind of biological materials. So we have bacteria, but also a lot of fungi and yeast. Bacteriophages are becoming of more interest during the last years. And we also have human, animal, and plant cell lines and DNA samples. And in general, we test viability and purity of the biological material. And we do that by controlling the growth or the cell division. And we observe, we check macroscopically and microscopically the purity and also the main biological fact. And for example, for human and animal cells, you also check for potential contamination with mycoplasms. So that are the general tests, and they take up from 2 days up to several weeks, depending on the growth behavior of the material. And when everything is fine, we assign the deposition number. And for the Budapest Treaty deposit, the depositor gets a certificate for receipt and viability. And with that, he can start the application process for the patent. And it is very important that this deposit is before the patent application. So that is clearly regulated by patent laws. And of course, we also need to preserve the material. So that is stored in a genetically unchanged way. And usually we also propagate the material and we produce, at least for bacteria and fungi, we produce our own set of samples. So we store them in glass capillaries in liquid nitrogen, that you can see in the photos in the bottom. And for cell lines, we will take the samples of the depositor and store them in cryotubes in liquid nitrogen. And if possible, we also produce a second batch of a sample where we use freeze drying of the microorganisms and produces glass ampoules with lyophilized material. And this is stored in either liquid nitrogen storage tank, or we are about to use this automated storage system. So this is just in the beginning for our collection. And we also test viability and purity at certain intervals. And the ideal time scale would be, of course, directly after preservation, then after 1 year, after 5 years, and then every 10 years. So that would be the best way. But yeah, we have to, like, since we have already a lot of material, we have to see that we can manage this. And yeah, just as an orientation, there's also a fee for the patent and safe deposit. So for the patent deposit of microorganisms, it is 900 euro. For the accession of cell lines is 1,600. The furnishing is 140 euros. And the safe deposit is 330 euros. And then we have to pay a yearly fee of 100 euros per sample. And yeah, so of course we also provide samples on request, but following the rules of the Budapest Treaty, which means that we provide samples to the depositor or to third party on behalf of the depositor, or also to patent offices, and so other legally entitled third parties can apply for biological sample or using the regarding patent office. So if you, for example, have a European patent that is published, then a third party could apply for a sample, filling a form at the European Patent Office, and they authorize us to release a sample to this third party. So the depositor is informed about this furnishing, but he can't legally prevent this in this case. In contrast to the safe deposit, where the full control is in the hand of the depositor. So yeah, with that, I hope I could give a short overview on patent deposit and the Budapest Treaty system, and I'm open for questions.
Okay. So thanks a lot, Felicitas. It is very interesting, also because we are mostly interacting quite a lot with you, either taking strains or depositing. So I think there can be some questions from here in the audience, also the people connected. Well, I would start with one, because, you know, it's very clear the difference between the two, the safe and the patent deposit. I'm wondering if there is a case where maybe you start with a safe deposit and then you consider that there might be a patent deposit for the same strain. Is this possible, of course, and how is the procedure followed? You need to start from scratch, there is a movement from one to the other. Maybe it's a particular question, but it can be relevant.
Yeah, yes, thank you for this question. So yeah, actually that is happening quite frequently. So people start with a safe deposit because maybe they also don't know yet what will happen with the sample in the future. And then it's not a problem to transfer this kind of deposit into a patent deposit. And the important thing is here also that the deposit date is really the first day we receive the sample, also when it was a safe deposit. And then the notification on this transfer from safe to patent, this has to be before the patent application, but this is really possible and not a problem.
Okay, thank you. Other question? Diogo from Portugal.
Hello, good morning, Felicitas. Good morning, everyone. Thank you very much for accepting our invitation. My question is regarding with someone's just submit a strain to the culture collection to be deposited as a patent. Is this strain available to be commercialized for other research, or it just under protection during the 30 years?
So yeah, like I think there are different sides of the thing. So first, the patent deposit is itself is just the deposit, and then the rights to use it. It lies either like with a depositor or with a patent holder. Yeah, that could also be different persons. So the depositor could sell the rights to use the strain also to another person or like another party to use it in a patent application. And also this rights for the patent, of course, could be sold. So for example, for commercial use. So if someone has an interesting invention and has also the interest to make money with it, of course, this patent and the application in it can also be commercialized. And but the deposit itself, this is really only used for the disclosure of the patent. So for example, if someone wants to prove that the patent is correct, so or if or want to I don't know, in this whole patent process, other third parties have the chance also to check if another patent was interfered. So this could be, I think, could involve really complex rights. But in general, it would be possible that like a commercial product is is the outcome of the whole thing. But like the patent deposit itself is just a small part in this whole process.
Okay, and it's like if someone finds in the genome of one of the strains that is under submission of a patent, find a novel enzyme that you like to discover. So someone can buy that strain and to explore that enzyme that was not included in the patent because it's a different enzyme, for example.
Yeah, this might be different in different country and how also you so in this example I showed earlier, it is said that the material, at least we give, like for under the Budapest Treaty, must only be used for experimental purposes. So if you then want to commercialize something, then you would need to contact maybe the owner of the strain or the holder of the patent. But that are not that's not us in this case. So they need to contact the person who has a right to use the strain. So we only have the right to use it under our deposit rules. But in general, it could be that the same strain has different interesting properties and is used by different also stakeholders, and that we have different patents. But but if you receive a strain from us first, you can use it for experimental purposes. But if you want to use it for another, like for a commercial purpose, then you have to gain the rights to use it for this purpose.
Thank you very much.
Thank you. Yeah, maybe Lorna and then Michelle.
Yeah, good morning, everyone. Thank you, Felicitas, for this presentation and this for great information for us and also for Melania about Nagoya Protocol. I want to know the process from the accession form to storage of strains, for example, of fungi and bacteria, how long it takes this process. And for example, for me, I am from Tunisia, I'm from North Africa, I'm not from European country. And another question, if, for example, the same strains, I have a patent like national patent, can I have with this same patent deposit at Budapest and have it international patent or not?
So for the first question, so about the procedure, it depends a bit on the strain. So if it's just, for example, Lactobacillus plantarum that is not somehow restricted and also easy to cultivate, then usually like this whole process can take like 1 to 2 weeks. So like we check the form, we receive the material, we check the material, and then we issue the number. This can really be done in about 2 weeks when everything is like easy and clear. Of course, like also regulations are growing. So and also when we have like restricted strains when it comes to, for example, animal pathogens or plant pathogens, or yeah, other restrictions, then we sometimes need to apply for a permit. And so this can also, yeah, like slow down the process. But like in general, like up to risk group 2, everything is possible. So we are allowed to work with plant pathogens and also animal pathogens. So that's so like it's possible, and we also receive deposits from all over the world. So we have also experience receiving samples from like from African countries or also from Asia. Yeah, so from all over the world. But of course, like some regulations may slow down the process a bit, but it's really different. And for the second question was if we can have an international patent application. Yeah, I think that's exactly the sense of depositing in one IDA. So if you deposit, yeah, for example, with us in Germany, you can have an international PTC patent application.
Okay, thank you. Thank you.
You're welcome.
Yes, thank you for this very interesting talk. I had one question regarding the storage conditions, because you mentioned that there are only 2 options: dried material, you keep it dried, or in liquid nitrogen. Correct? But what happened? Do we need to test these conditions before shipping our strains to DSMZ, or are you testing these storage options yourself? And what happens if it's not possible to keep fungi, for instance, in neither of these conditions?
Yeah, so it has to be possible to preserve the material. So we can't accept material that is not preservable because this is just not tenable. But unusually, like, of course, usually people have experience how to preserve their material, but we check it in general. So like we usually test our standard procedures, and like most organisms are preservable, at least in liquid nitrogen. Of course, we have some organisms that are not so easy to preserve with lyophilization. For example, some of the fungi, and also some like difficult bacterial strains. But for at least the liquid nitrogen preservation is possible in like almost all cases. Sometimes, like for the plants and animal cell lines, we also take material from the depositor, and we have some really exemptions where we also accept microorganism material that is already preserved and stored directly if the preservation protocol is really that complicated that we don't manage. But these are really like I don't know, not even a handful of examples. So that's really usually it's possible. But of course, like there are still some also microorganisms or maybe plant cells that you can't not preserve, then it's getting difficult. Actually, I'm not sure if other IDAs may accept also non-preservable material. I would need to check that.
Because some, let's say, symbiotic fungi, for instance, for plants, are difficult to keep as freeze-dried material or liquid nitrogen. So we need to maintain some living material some prejudicial material, but it's very time-consuming, I guess.
Yeah, so like we usually don't accept the material in this case, but there might be other AZAs who do that. But like there is information on the WIPO system website where all biological material accepted by the different IDA is mentioned. So one you as a depositor, you could check which IDA accept which material. So that would be the option, but it's really getting difficult because we have to guarantee the storage over 30 years more or less, and if you have to handle it and also pass on frequently, we couldn't guarantee that it is not changed in this period. So this is really difficult then.
Okay, thanks a lot. So maybe I think we should close the first part of the seminar. And we thank you really again a lot because this is extremely important and useful for us. So we thank you for your time. Of course, if you have time also to attend the next part of the workshop, you are welcome, depending on your schedule. And so we have a break when we start. When we start 11, I don't know. 11. Okay. Yes, so 10 minutes, 10, 12 minutes of break.